Good evening. Can you hear me? Uh, Please show me if you can see my screen. Is it shared or not? Okay, thank you so much. Thank you for your answer. Good evening, everyone. Thank you all for being here today. It's a pleasure to present on a topic that is fundamental to clinical laboratory operation, method validation in GCLP lab under FDA supervision. Let me introduce myself. I am Shahrzad Soori, and today I will walk you through the key principles, process, and regulations surrounding method validation. This presentation is designed to help you understand not just the technical aspects, but also the broader implication of adhering to GCLP standards under the FDA's supervision. Why is this topic important? Because the accuracy okay, now it's better. Because the accuracy, reliability, and compliance of laboratory data directly impact drug development, patient safety, and regulatory approvals. By the end of this session, you will gain insight into why method validation is not just a procedural necessity, but a cornerstone of quality and trust in lab operations. First, I'd prefer to explain about method validation in GCLP lab. In pharmaceutical companies, before a drug or product is sent to market, several critical steps ensure its safety, efficacy, and compliance with regulatory standards. One of these essential steps is method validation, which happens at multiple stages. First, initial validation in the pharmaceutical company itself. When a drug or product is developed, the manufacturing company designs and validates analytical methods. These methods are used to test the product's quality, for example, for purity, potency, or stability. This step ensures that the methods are robust and reliable for routine quality control during production. Second, revalidation in a GCLP lab. It's really important. GCLP lab is a laboratory that works under supervision of FDA. Once the product enters clinical trial, the same or similar methods are sent to GCLP compliant laboratories to validate under clinical trial conditions. GCLP lab specialize in handling clinical trial samples and must confirm that the methods work as intended in a controlled, regulatory compliant environment. This step ensures that the methods are not only scientifically valid, but also meet FDA standards for use in clinical research. FDA approval is the third step. After method validation in the GCLP lab, the data generated using these methods is submitted to the FDA for review. The FDA examines the robustness of the validated methods, compliance with GCLP and regulatory guidelines, whether the methods ensure reliable results that support the drug safety and efficacy. A simplified workflow of my explanation is first, pharmaceutical company that develops a product and validates analytic methods. Second, GCLP lab independently revalidate these methods for clinical trial use. And third is FDA review, approves the drug based on validated method and clinical trial results. So, then I can start with my presentation now. Okay, so what is method validation? I should say about method validation now. Uh, okay, let me check, please. Yes, it's fine. Okay, no problem. Okay, thank you. Okay, thank you so much. Introduction. What is method validation? Method validation is the backbone of laboratory accuracy. It involves verifying that a given method consistently produces reliable and reproducible results, whether it's measuring a drug's potency or identifying a biomarker. The process ensures that the method aligns with its intended purpose. Imagine testing a life-saving drug without validating the method used. The risk of incorrect result could lead to significant consequences, delayed approvals, patient harm, or even failed clinical trial. And now I should say about the GCLP lab. GCLP or good clinical lab practice bridge. I don't know. Arman is here or not? I want to ask some question of you. Okay, I cannot see him. So, okay, no problem. GCLP or good clinical laboratory practice bridge the rigorous standards of GLP with clinical trial requirements. Unlike standard laboratory practices, GCLP ensures that all data generated during clinical trials meet stringent quality and ethical benchmarks. It's the gold standard for clinical labs. And what's the FDA roles in this field? The FDA oversees compliance with GCLP and method validation standards. Its goal is to ensure that all laboratory results used for regulatory submission Sorry, I think my PowerPoint has a problem. Okay, no problem. It's okay. Uh, its goal is to ensure that all labs results used for regulatory submissions are reliable and reproducible. Without meeting FDA expectations, labs risk producing non-compliant results that can detail clinical trial and regulatory processes. Uh, about importance of method validation, I can say why is method validation important? The first thing that I should say is that method validation ensures accuracy. It means that accurate methods yield true results. For example, in clinical trial, inaccurate results could lead to unsafe or ineffective treatments. So it is the most important thing that a method validation can help us to produce a test, lab test, or maybe a product for medical field that has stability, is reliable for using by patient, and it's FDA approved, and we can use it without any problem. The second one is compliance. Regulatory standards demand robust validation to ensure that lab results are legally defensible. And third one is patient safety. Unvalidated methods can lead to misdiagnosis or harmful drugs reaching the market, putting lives at risk. And the fourth one is reproducibility. A validated method ensures that any lab replicating your process achieves the same reliable results. Um, uh, Sorry, I'm so stressed because for tonight I couldn't find a babysitter, and my friend now take care of my baby, and he's really restless. I'm so sorry if you hear the my baby sound. I'm really stressed. I'm so sorry. Uh, so okay. Uh, Key component of method validation. Let's explore the components one by one, emphasizing their role in achieving robust validation. The first one is specificity. Can the method distinguish the analyte of the interest from other substances? For example, does it correctly identify a specific drug in the presence of impurities? It's really important that a drug or product has a specificity, and it should be enough for using in industry, medical industry. The second one is accuracy. How close are the results to their true values? This ensures that the method measures what it claims to measure. And the third one is precision. Precision focuses on reproducibility. For example, if a method is applied multiple times under the same condition, do the results remain consistent or not? It's really important. The fourth one is linearity and range. Linearity, results should be proportional to the analyte's concentration. All of the uh, lab colleagues notes that about the linearity, and it's really important. Method validation probe the linearity about any um, lab result for any drug or any lab tests that we want to validate it. About the range, defines the boundaries without which the method performs reliable. The fifth is limit of detection. That here we have two important measurement. It's LOD and LOQ that is really important in any method validation. LOD is the smallest detectable amount, and LOQ is the smallest quantifiable amount. These parameters are critical when dealing with trace impurities or biomarkers. And the sixth is robustness and ruggedness. Robustness, about the robustness, we can say measures method performance under the minor variations. The variation is really important, and many factors can affect this item. And about ruggedness, evaluate consistency across labs, equipment, and operators. These factors can affect this item. For example, a drug testing method might fail robustness testing if it produces varying results under a slight pH change, highlighting areas for improvement. The next slide is about FDA guidelines and GCLP requirements. I can say that core FDA expectations of any validation, any method validation that we are using in GCLP labs is that the first one, develop clear validation protocols, and the next one is maintain evidence of reliability through validation reports and SOPs. The SOPs or SOPs, some colleagues in the lab say SOPs, are really important in conducting the lab tests and any procedure in GCLP labs. And the last one is conduct continuous monitoring and periodic revalidation. Integrating GCLP and FDA guideline, this item, integrating GCLP and FDA guideline. GCLP emphasizes ethical sample handling and data integrity, while the FDA focuses on regulatory compliance. Together, they form a robust framework for laboratory operations. Practical implementation in labs. A step-by-step validation workflow include the first one, define the method's objective. The next one, develop the protocol, selecting appropriate reagents and equipment. Third one, perform validation tests, assessing accuracy, precision, and others that I mentioned before. The fourth one is analyze data to confirm reliability. And five is document everything for future reference and regulatory audits. We have important rules in GCLP labs that we should maintain all the documents for any clinical trial project at least for 20 years. At least for 20 years, we had to maintain and retain these documents because of any consequence of any bad result when the pharmaceutical companies start to sell this product in the market, maybe it will have some problem or maybe side effects. So we have to retain all the document for at least 20 years in GCLP lab. It's really important. And Data integrity is really important in validation. Validation is not just about results, but also about traceability. Proper documentation ensures that data is defensible during audits. For all the GCLP lab, they should have audit every 2 years. This audit is related to FDA, and all the GCLP labs, if wants to continue their works, they had to pass this audit. And if they have any problem or maybe any non-conformances, they have to solve it, and in next audit it should be solved. So the audit is really important in a GCLP lab, and every 2 years all the GCLP labs should pass this audit. And they have another audit that is internal audit, but here just I explain about the FDA audit. The last slide of my presentation is about a conclusion and question. Method validation ensures accuracy, reliability, and compliance. FDA supervision protects patient safety and drug efficacy. Continuous validation builds trust in laboratory data. And I should say thank you for your attention. I hope this presentation has provided clarity on the importance of method validation in GCLP labs under FDA supervision. And I'm happy to answer any question you may have. Okay, no problem. Do you have any question or not? No. Okay, thank you so much. Thanks for your attention. Thank you so much, Niku. I wish all the best for you. Good night. Bye.